I'd like to thank the organisers for the invitation to speak to you this afternoon, and my talk is entitled Adapting Viral Diagnostics to New Variants of SARS-CoV-2. Here are my disclosures. So since the beginning of the SARS pandemic, we've noted emergence of variants towards the end of 2020. We now have three main new variants of SARS-CoV-2, the South African, Brazilian, and UK variants, otherwise known as 501Y V1, V2, and V3. These new variants are characterized by the fact that they have multiple mutations in spike, and especially in the receptor binding motif, which is part of the receptor binding domain in the S1 part of the spike protein. And of course, the spike protein is sitting on the outside of the virus and engaging with ACE2. Here you can see a schematic diagram, a ribbon diagram at the bottom, which shows spike receptor binding motif in conjunction with the ACE2 receptor. And you can see that there are three key interacting residues amongst a number of others, but these are the three residues that are found in common in at least two of the emerging variants. You can see position N501 here at the top right. This makes a key contact with ACE2, as does K417 and E484. And these are mutated in the variants to N501Y, E484K, and two different mutations of 417 are observed, K417T and K417N. On the bottom right here, you can see from the Wiesler lab some data on interferometry, where they have measured the binding between the receptor binding domain purified protein and human ACE2. In figure A, you can see the on and off rates at different concentrations of the wild type receptor binding domain. And you can see that there is occupancy for a considerable period of time. At the bottom, you can see what happens when you have an N501Y mutation. The on rate is a little bit slower, but the off rate is also considerably slower. So this translates into greater occupancy time. And that is likely important for this adaptation the virus is making in terms of hanging on for longer to the ACE2 receptor. N501Y is also known to be an escape mutation for certain antibodies that we produce. And therefore, there is an unknown interplay between receptor binding and escape from antibodies. This set of structures is illustrating the different variants that I've been describing. On the left here is 501Y V1, in other words, the UK variant. And you can see here the N501Y on the top in the receptor binding domain, as we've just seen. In addition, other key mutations are in the S1 fragment, the delta 6970 deletion, which you can see there, as well as the Y144 deletion. Now, these are really important because the Y144 is in a known antibody binding site for the 4A8 antibody, and delta 6970 removes two amino acids in an exposed loop, potentially leading to increased infectivity based on some data from our lab. The delta 6970 is not known to be an antibody escape mutation. There are a number of other residues mutated, including A570D, and of course the 681, P681H near the furin cleavage site, there you can see in orange, and a number of other S2 mutations in red. Moving on to 501YV2, in other words, the South African variant, you can see here that there are a number of mutations in common, for example, the 501Y. And one thing I haven't mentioned is, of course, that the UK variant on the left, the V1, has now been shown to be developing a cluster of infections containing the E484K mutation. Moving on to the right-hand side, you've got 501YV3, in other words, the Brazil origin variant. And you can see there are a number of different mutations in addition to 501 and 417 mutations. For example, L18F, and you can see a number of others there in the diagram spread throughout S1 and S2. So the next question is, why have these variants why have they arisen? So here is a case that we recently published of an individual treated for chronic COVID-19 infection spanning 4 months, which unfortunately resulted in death. The individual had an underlying lymphoma and was admitted to hospital. And you can see in this figure on the left that the CT values from nose and throat swabs was consistently elevated. And you can see the clinical course there with the patient being admitted with breathlessness and cough, requiring CPAP or positive airways pressure, and then eventually intensive care unit, and then intubation, but unfortunately progressive disease and death. During that admission, there were a number of treatments administered, including remdesivir, but also 3 units of convalescent plasma. And on the right here, you can see the variant frequencies in this individual between days 82 and 100 of the illness. And you can see big fluctuations in virus populations, almost looking like there are competing virus quasi-species. But in particular, we documented the real-time emergence of the Delta 6970 mutation in real time, suggesting that this is a mutation that can emerge during chronic infection, consistent with the fact that we're seeing this, as well as many other mutations, arising in the variants that I've just shown you. So what are the implications of all of these variants for diagnostics? Well, for the 501YV1, we have undertaken in the UK a number of tests using the standard platforms, including, for example, the Panther setup, and there doesn't seem to be a significant change in the diagnostic performance of our laboratory tests. For the 501 V2 in South Africa, we can see that a number of platforms have been tested, including the Abbott M2000, Roche 6800, and Cgene CFX, as well as the Gene Expert, and these all show a maintained performance against the circulating variant in South Africa, so that's reassuring. As yet, I'm not aware of any data for the Brazilian variant 501YV3 on diagnostic nucleic acid testing performance. Here are some tests that may be affected based on the primer designs. The first is the Acula SARS-CoV-2 test by Mesa Biotech, the linear COVID-19 assay by Acula, and as I said, these may be affected because of S-gene targets. And this is likely due to S-gene changes in the B117 variant in the UK, also known as the 501YV1 variant. But the interesting thing is that we actually gained a diagnostic test in some regards by the emergence of these new variants. And this is because of that deletion at 6970 and spike that I talked about earlier. The phenomenon of S-gene target failure in the context of B117 was one of the clues that people had in the UK that there was something different and a different strain emerging in the UK. We used the Thermo Fisher TaqPath testing platform in many of the centralized laboratories that have been testing community specimens for SARS-CoV-2 in nasal swabs. This test uses 3 gene targets. One is in the S gene, and this covers the 6970 deletion region. The other is in the N or nucleic capsid, and one in ORF1AB. As I said, it's commonly used in community testing. And when they retrospectively looked at over 31,000 samples from the community and looked at the S-gene target failure status, they realized that nearly all of the Delta 6970 sequences were cases of S-gene target failure after sequencing, compared to very few sequences which had S-gene target failure in the absence of deletion. So this suggests to us that there are a number of, that the S-gene target failure was due to the deletion 6970. This graph shows you what was happening over time in the proportion of tests that showed S dropout because of that 6970 deletion. And that is shown in black. You can see that the proportion of sequences shown to be B117 mirrors this beautifully in dashed red line. And the other 6970 deletion lineages, which are present in the UK, for example, the 439N439K plus 6970, have remained at relatively stable proportions during that time and even falling off slightly. So you can see a very nice correlation between the emergence of B117 or the 501YV1 variant in the UK and the S gene dropout signal. So looking more closely at the UK variant, here you can see the nucleotide and amino acid changes spread across the virus, in ORF1ab, in spike, there are 8 mutations. There's also, there are also mutations in ORF8, including a stop codon, and there are changes in the nucleocapsid. This variant is now present in more than 80 countries, as many of you know, and we know that it likely has a transmission advantage and potentially increased mortality, although those data remain to be verified. We have done some work on whether this new variant is going to be susceptible to vaccine sera generated by the Pfizer vaccine or the BioNTech vaccine. And we can see here that the amount of serum required for neutralization of pseudotyped virus is considerably higher for the B117 variant. And so we found a fold change around between 2 and 3 for the B117 variant relative to wild type. This appears relatively modest, but this is a single round assay and it's likely to be much greater when you look at multiple rounds of infection. We've also tested a number of convalescent sera and there is variable response. This diagram here shows you a representative example of somebody who has a significant fold change and the B117 is less well neutralized by this patient's sera as compared to wild type in blue. So going back to the 6970 deletion, of course, the B117 is one virus that contains this. On the top left, you can see where this deletion is. It's there in blue on an exposed loop that you can see, and you can see where it is in relative to the 488 epitope in purple. On the bottom shows you what happens when the 6970 deletion takes place. You can see in green, this is the post-deletion structure, and in blue is the pre-deletion structure. So there is a conformational change that's predicted, but this hasn't been proven through structural studies yet. And in the middle, you can see now the phylogenies of 6970 deletion containing viruses. In pink at the bottom, you can see the very large cluster of B117 containing viruses, and in the gray bars show you the fact that this has a 6970 as well as a 501Y mutation. If you move up that tree, you can see just above the pink is a group of viruses that has 501Y in the absence of the 6970. And further up, you can start seeing viruses that have the 453 and 6970. Those are from Denmark, as you can see in green, and we know that's the mink-related lineage. Interestingly, the 6970 deletion came after the 453 was acquired. As we go up, you can see the B1.351, which is a 501Y containing lineage. Of course, this is the South African variant. And then further up tree, you can see the 439K containing viruses that emerged initially in Scotland on their own at the top of the tree. And then you can see further down that they have acquired a 6970 deletion later on, and that now is dominating the 439 lineage. So this is just showing you that 6970 deletions have come up independently and in combination with other receptor binding domain mutations such as 439, 453, and 501. So this is an important deletion to consider. But we should be cautious against assuming that all S gene target failures are equal to B117. Here's some data from the Czech Republic and Slovenia, recently pre-printed, showing you that the B117 has similar viral loads to a lineage that contains N439K and the 6970 deletion that I showed you earlier that arose in Scotland, we think. Certainly the 439 arose in Scotland. Here is a phylogenetic tree on the right showing you the acquisition of 439 in the United Kingdom, and then the deletion 6970, which came after that. And of course, so this is an example of why you cannot use the S gene target failure in the TACPath assay as a proxy marker for B117. You need to do sequencing validation to understand what the proportions of different viruses are in your country or location. So it can be used as a surveillance tool, but it must be backed up by sequencing as well. Here's another case as to why you can't use the S gene target failure as a proxy for B117. This B1.375 lineage in the United States was recently described as well. And this is a lineage with only 6970 and a few other mutations. But again, the only spike mutation is delta 6970. It is not a B117 lineage. What about lateral flow antigen tests? Well, the ANOVA, SureScreen, Abbott PanBio, Roche Biosensor, and Fortress antigen tests have been verified by Public Health England on the UK variant, and the performance is not affected by the new variant, which is reassuring. And this is because most of these tests are looking at the C terminus of the N gene. We don't have any data on South African variants yet, and that it needs to be done in addition to the 501YV3 variant. What about rapid nucleic acid testing platforms? The Samba2 platform that we validated last year appears not to be affected. This is a test that gives you a turnaround time of 1.5 hours. Unfortunately, it only does one sample at a time, but multiple machines can obviously increase your throughput. This test uses the E gene and the ORF1A, and therefore is unaffected by the B117 variant. What about antibody testing? Well, we showed last year that point-of-care rapid antibody kits, in particular two, the SureScreen and the Prometheus tests, were not affected by the D614G mutation. As you can see here, there are a number of our sequences in the panel were G at the 614 position, and this is middle of last year, but we did have some Ds at that position. And if you look on the right-hand side, you can see the results from D614G containing virus sera of patients who had been infected with those viruses and recovered. You can see that there's a very nice correlation between the intensity of those bands in the POC testing kit and the intensity over time. So that's an example of how a mutation in spike did not affect the rapid tests. We still haven't done those assessments for the B117 variant, but those are ongoing. What about lab antibody testing? Of course, well, S versus N-based tests may give different results. There's very little data on these, but we do urgently need to do these assessments because going forward, we will need to do accurate serological tests on populations in order to be able to track infection or past infection in those individuals and potentially try to differentiate it from vaccinated individuals. So in conclusion, I have shown you that the new variant can be detected through S gene target failure in nucleic acid testing. However, we need to be aware that there are other strains with the 6970 deletion, for example, N439K circulating variants. We think that some other nucleic acid testing platforms may be affected, but more needs to be done. There is very little data on South African Brazilian variants or 501Y V2 and V3 variants. And antibody tests need urgent further evaluation. So there is a real need here to improve the amount of information we have. I'd just like to end by acknowledging the individuals on this slide for all their hard work. Thank you very much.